Welcome, dear students. Uh, these days I'm dealing with a particular topic, that is blood groups in human beings. Now I'll be dealing part 2. In part 2, I'll be dealing ABO blood groups. The ABO blood group in human beings was first discovered by Lance Tinner in the year 1900 on the basis of A, B, O blood groups. Human beings have been classified into 4 groups. These are A blood group, B blood group, AB blood group, O blood group. Human red blood cells, which you call RBCs and also called erythrocytes, contain different types of antigens on the outer surface of their plasma membrane, which give rise to different blood group system on the basis of different types of antigens present on the surface of the RBC, red blood cells or erythrocytes. They are grouped in different groups. Like you see, A blood group, human RBCs having antigen A. In B blood group, human RBCs having antigen B. AB blood group, human RBCs having both antigen A and B. And O blood group, human RBCs having neither antigen A nor antigen B. Now you see the antigen antibody interaction, how these antibodies are formed in reaction of the antigens. Generally antibodies are produced in response to foreign antigen. It is the system, biological system. But antibody A, which you call anti-A, is naturally produced in blood serum of blood group B. Blood group B, which you when you analyze the serum, that will be having the antibody A. In the serum of blood group B people, you are having antibody A, anti-A, which you call. Is produced in blood serum of likewise group B, and antibody B is produced in blood serum of blood group A. Blood group A serum is having antibody B. Naturally it is produced. But blood serum of blood group AB does not have antibody A or antibody B. And blood serum of blood group O people contain both the antibody A and antibody B. So this is the situation, that O blood group in its serum they are having antibody A and antibody B both. And in case of AB blood group, in the serum you don't have antibody A or antibody B. And likewise in A, blood group people, the serum is having antibody B. And likewise, in B blood group people, the serum is having antibody A. So on the basis of this antibody antigen interaction, this agglutination, the coagulation, or you can say blood clotting, is happening because of this. So the proper blood group has to be tested at the time of transfusion. A may have. I'll be discussing later about the blood transfusion. So hence it is very important to know the exact blood group of donor and recipient at the time of blood transfusion. If incompatible blood is transfused, for example, if blood from group B, which is having antibody A, is transfused into group A, which is having antigen A, then antigen antibody interaction takes place, causing agglutination of RBCs, which will be fatal to the person. So we must try to understand, we must test it, then only you go for blood transfusion. So try to see the compatible blood group for blood transfusion in human beings, the ABO blood groups. You see the blood group, the antigen on RBC, red blood cells, the antibody in the serum, the compatible transfusion from. You see here, if a person is having blood group A, of course he is having on his red blood cells the antigen A. Of course in his blood serum he is having antibody B. So he can receive blood from only A, which is compatible. A blood group he can receive, or O blood can be, theoretically O blood can also be transfused in A blood group people. Similarly, in case of B blood group, B is having antigen B on its RBCs, and in serum antibody A is present, so you can only transfuse B blood group into B as well as O. Theoretically, O can also be transfused in B. Now you see AB blood group. AB blood group is having on its RBCs the antigen A and antigen B both, and because of that in its serum there is no antibody, neither A nor B. That is why it cannot accept it can donate blood to only AB, not to any, but it can receive, because of this situation, no antibody in the serum. Because of this situation, this can receive blood from any group. So this is beneficial. The AB blood group people, they can receive blood from A group, from B group, as well as, of course, the AB is the similar from that, as well as from O group. So that is why, because of no antibody is present in the serum and A and B both antigens are present in the RBCs, this is called universal recipient, because you can transfer any blood group into AB. And reverse to that, the O blood group is problematic Of course, uh, you don't have antigen. You don't have antigen in case of O blood group serum, RBCs, but in its blood serum, O blood group blood serum is having antibody A as well as antibody B. So it cannot receive blood from any except its own. That is why you call universal donor, because it is having antibody A and antibody B both in the blood serum, but there is no antigen in the RBCs. That is why you can donate O type to any group, A, B, AB, to any. That is why you are seeing here in all the groups O, you can transfer O blood group to any. So that is why you call the O blood group as a universal donor. So this is all about the blood groups in human being part 2. Thanking you very much.